Hello students, last class we have seen about the body composition and how it occurs in the body, what are the various components of body composition. So, today we will see what are the methods for assessing the body composition. So, in physical fitness, body composition refers to the quantifying of different components or the compartments of a human body. The selection of compartments varies by model, but may include fat, bone, water and muscle. Two people of the same gender, height and body weight may have completely different body types as a consequence of having different body compositions. This may be explained by a person having low or high body fat, dense muscles or big bones. Now, compartmental models, the body composition models typically use between 2 to 6 compartments to describe the body. Common models include 2 compartment which includes fat mass and fat free mass, then 3 compartment which includes fat mass, water and fat free dry mass, then 4 compartment includes fat mass, water, protein and minerals, and 5 compartment fat mass, water, protein, bone mineral and non-osseous mineral content, and 6 compartment which includes fat mass, water, protein, bone mineral content, non-osseous mineral content and also glycogen. As a rule, the compartments must sum to the body weight. So, the proportion of each compartment as percent is often reported, found by dividing the compartment weight by the body weight. Individual compartments may be estimated based on the population averages and measured directly or indirectly. Many measurement methods exist with varying level of accuracy. Typically, the high compartment models are more accurate as they require more data and thus account for more variation across the individuals. The 4 compartment model is considered as a reference model for the assessment of body composition as it is robust to most variation. Each of its components can be measured directly. Coming to the measurement methods, there are different main body composition analysis tests on the market and each varies on the method and the accuracy used. So, here we find the different methods like the hydrostatic or underwater weighing, air displacement BODPOD, DEXA or dual energy X-ray absorptiometry, then isotope dilution analysis, body mass index, MRI which is magnetic resonance imaging, NMR spectroscopy, then PET computed tomography, PET or MRI, bioelectrical impedance analysis, body volume index, skin fold test, ultrasound, quantitative magnetic resonance, circumferences and other simple measurements. So, we have about 14 types of different measurements which we will see in detail. So, a wide variety of body composition measurement methods exist and the gold standard measurement technique for 4 compartment model consists of a weight measurement, body density measurement using hydrostatic weighing or air displacement lithosomography and total body water calculation using isotope dilution analysis, mineral content measurement by dual energy X-ray absorptiometry which is DEXA. However, it is also common to use a DEXA scan alone and refer to this as a gold standard. These claims are somewhat dubious since measurements methods vary significantly from study to study. In practice, the measurement methods used will be trade off between cost, availability and accuracy. Coming to the hydrostatic method, hydrostatic or underweighing water is a form of densitometry that uses the body weight on land and in water along with water displacement to estimate the body composition. This method is based on Archimedes principle. The buoyant force on a submerged object is equal to the weight of fluid that is displaced by the object. Muscle does not weigh more than fat, but it is denser and takes up less volume per pound. So, in other words, muscle sinks and fat floats. So, a person with higher percentage of muscle will sink, and this method is based on Archimedes principle. The buoyant force on a submerged object is equal to the weight of the fluid that is displaced by the object. Muscle does not weigh more than fat, but it is denser and takes up less volume. In other words, muscle sinks and fat floats. So, a person with more muscle mass will weigh more underwater than a person with high percentage of body fat. So, when performing underwater weighing, a person is required to expel all the air from the lungs during the test, as any extra air will impact water displacement. So, the test is typically repeated 3 times and the average underwater weight is used. This method is praised for being pretty accurate and has a margin of error of around 1 to 3%. The next method is air displacement lithosomography. The body composition measurement with air displacement lithosomography or whole body air displacement lithosomography technology. Air displacement BODPOT, the air displacement works similar to water displacement except an air controlled chamber resembling an egg shaped pod is used. This is called BODPOD. So, a BODPOD uses a mechanical diaphragm to create small volume changes in the confined and controlled space, then measures pressure responses. A person's weight, age, gender and height are recorded before the test and the chamber is calibrated. And then using the basic understanding, density equals mass over volume. A person's mass is recorded with their body weight. Volume is measured using air displacement in the BODPOT. Then, since both fat mass and fat free mass have known densities, the BODPOD can estimate how much fat and how much fat free mass a person has. So, to get BODPOD test, there is a need to find a facility that will perform the test for, and this is not an easy feat. Most BODPOD are found in medical and health facilities and are not widely available to the public. The margin of error in BODPOD is similar to underweight water weighing, which is 1 to 3%. Then the DEXA scan. This DEXA scan is the most accurate and advanced form of body composition analysis that is available. It uses X-ray technology to scan the body and provides a detailed assessment of how much muscle mass and fat mass down to the pound and exactly where the fat and muscle is stored in the body. For example, if the right arm has 0.5 more pounds of muscle than the left arm, a DEXA scan will identify this. It is so accurate. This also means DEXA scan will differentiate between subcutaneous and visceral fat. It can even tell bone density. The margin error for DEXA scan is just 1.6%. DEXA scans to be the most useful, easy, inexpensive test for helping diagnose osteoporosis. Then isotope dilution analysis. Total body water may be effectively measured using isotope dilution analysis of deuterium oxide, and the total body potassium. So, potassium 40 is the naturally occurring radioactive isotope found in the intracellular water in the body, but it is not present in stored triglycerides, which is a component of the fat. So, whole body counting can measure the amount of potassium 40 which is present in the body, and a quantity called total body potassium can be measured. This can be used to estimate fat free mass directly, and it has most been replaced by newer, more accurate techniques such as DEXA. Then coming to the body mass index, then body mass index is used to work out if you are healthy weight. It is calculated by taking a person's weight in kgs and dividing it by the height squared. The higher the figure, the more overweight and the greater the health risks. Quick and easy and with minimal cost, and it matters for more adults. There is a clear correlation between higher BMI and negative health consequences. As with any weight measure, reliable scales are needed, plus you need a tape measure to take the height. It can differentiate between fat and lean mass weight, and it is not very accurate for people who are elderly, pregnant or very muscular. Then we have the magnetic resonance imaging MRI. This MRI is only available non-radiation technique that allows for the in vivo quantification of total adipose tissue and its subdepots, subcutaneous, intramuscular and visceral adipose tissues. The MRI also allows for quantification of important fat free components, skeletal muscle mass which are present in arms, legs and trunk, and also in the specific organ mass. Now, organs such as heart, liver, brain, which have high resting metabolic rates relative to their masses, which is highly relevant to the investigation of the effects of weight perturbations, it may be gain or loss on the resting energy expenditure. So, MRI can also provide estimations of bone marrow adipose tissue. Limitations of MRI include high cost to acquire the scan, specialized post processing software that is required for tissue volume analysis by a highly trained analyst. Then participant requires to remain completely motionless during the scan, which takes about half an hour, and making it very difficult for young children, and it is also not feasible for persons with claustrophobia. So, it is used to investigate or diagnose conditions that affect the soft tissues such as tumors or brain disorders. Then NMR spectroscopy. NMR is a non-invasive technique and a standard non-invasive technique for measurement of intrahepatic lipid and intramyocellular lipid. So, extracellular lipid in the muscle fibers. So, you can measure the fat present in the liver and between the muscle cells and outside the muscle cells. So, NMR is a surrogate for liver biopsies to detect fat content in the liver. Physical activity and overall fitness have been associated with increased IMCL, and whereas EMCL is increased in persons with overweight or obesity. Increased EMCL and decreased IMCL in the vastus lateralis muscle are independently associated with arterial stiffness in the adults. The NMR metabolomics has been used in the identification of biomarker for cardiovascular diseases and risk stratification. It has also been used in cancer diagnosis and therapy. The NMR metabolomics has been used to investigate processes like transformation, progression, proliferation and metastasis in the cancer cell lines. Then PET or computed tomography and or PET and MRI. In the PET, a tracer, usually 18F fluorodeoxyglucose, is introduced into the body and accumulate in clusters of high glucose metabolism that can be measured and visualized in the PET scan alone or more commonly combined with computed tomography. Now, PET scans are used to identify highly metabolically active areas in the body. Recently, this technology became the gold standard for detection of brown adipose tissue depots in the humans. So, although the prevalence of brown adipose tissue in humans is estimated to be very low, only 2 to 7%, now studies have found that an inverse association between the brown adipose tissue and BMI in children and adults. The brown adipose tissue has been proposed to be protective against obesity or obesity related complications. And the biggest disadvantage of using PET CT for brown adipose tissue detection is significant radiation dosimetry associated with the CT scans coupled with very high cost. Now, recently PET MRI has been proposed as an alternative to PET CT, thereby avoiding ionizing radiation while allowing for studies in children as well as repeated measurements in healthy children and adult population. Further, PET MRI allows discrimination of the white adipose tissue and brown adipose tissue in children, whereas differentiating active and non-active brown adipose tissue depots. In general, PET scan may be used to evaluate organs or tissues for the presence of disease and other conditions. PET may be also used to evaluate the function of organs such as heart or brain. And the most common use of PET is in the detection of cancer and evaluation of cancer treatment. Then we have the bioelectrical impedance, which is another method to estimate the body water in the bioelectrical impedance analysis, and this uses the resistance of electrical flow throughout the body. The bioelectrical impedance is a highly sensitive to hydration status and water intake. So, drinking water dilutes the electrolytes in the body, making it less conductive, as does increasing bad body fat. By controlling hydration status or performing multiple impedance measurements, it is possible to estimate the body fat and other variables. The InBody developed the world's first 8-point tactile electrode system in 1996, a direct segmental analysis method that measures the impedance of 5 torses using multiple frequencies. Many bioimpedance analysis products they provide partial muscle fat mass measurements, but not impedance, especially in the torso. Recent advancements such as 8-point electrodes, multi-frequency measures and direct segmental analysis have improved the accuracy of bioelectrical impedance machines. So, these machines have found acceptance in medical, fitness and wellness space owing to their ease of use, portability quick measurements and cost efficiency. Aura Devices developed and launched a number of wearable bioimpedance trackers, including the first smart strap for Apple Watch that tracks body fat, muscle, lean mass and water levels, etc. Then we have another method called the body volume index. So, here this is a technique which is used to measure the body shape. Initially, the body volume index technology employed white light scanning machines to measure an individual's body shape. However, the recent technological advances in 3D measurements have enabled to measure the individual's body shape. So, these technologies now have been calculated by using images taken on smartphone. Two images are required to create an individual's 3D silhouette. By comparing this 3D silhouette with MRI data, body volume and fat distribution can be calculated. Then we have the skin folds. The body composition can also be measured using skin fold test, which is performed using measuring calipers, and this can be done in 9 steps. So, the skin fold tests are one of the oldest and most basic ways to measure the amount of body fat one has. Of course, because of the simplicity to this approach, it is also the most inaccurate, and this method uses the skin fold calipers to pinch the body in specific areas like the chest, thigh and abdomen and measure the skin fold thickness. This can be done through a 3-point measurement or a 7-point measurement that expands the test to other body parts, including the triceps, below the armpit, which is called the mid axillary, shoulder blade, subscapular and hip, suprailiac. These measurements are then combined with a person's age and gender and put into a specific formula to provide the percentage of body fat. Skin fold tests are limited because it only measures the subcutaneous fat, which is present just below the surface of the skin, and does not fully measure the body assessment. Full body assessment cannot be done by skin fold tests. Not everyone stores the fat in their body in the exact same way. It is also the subject to human error, depends upon the performance of the person who is testing. One can get skin fold test from many gyms and fitness centers, and the cost will vary. A common skin fold measurement is by using gun style calipers to measure the thickness of subcutaneous fat in multiple places of the body. This includes the abdominal area, the subscapular region, arms, buttocks and thighs. These measurements are used to estimate total body fat. Then we have the ultrasound. Ultrasound also has been used to measure subcutaneous fat thickness, and by using multiple points an estimation of body composition can be made. Ultrasound has the advantage of being able to directly measure muscle thickness and quantify the intramuscular fat. In the abdomen, the ultrasonography is a useful tool for quantifying both subcutaneous and visceral fat. The ultrasonography has many advantages over CT scan and MRI, as it is a non-invasive and does not utilize ionizing radiation, making it more accessible to the special population. Then we have the quantitative magnetic resonance. This quantitative magnetic resonance applies magnetic field to the body and measures difference in relaxation rates of hydrogen atoms within the fat versus the lean mass. It functions similarly to the magnetic resonance imaging, but instead of providing an image like MRI, the QMR gives the quantities of fat mass, lean mass and total body water. So, QMR is also widely used for body composition analysis of animals, including laboratory animals like mice and wildlife including birds. Circumferences and other simple measurements also can be used for assessing the body composition. So, this is done by assessment of somatic, that is skeletal protein, which is typically determined by simple measurements and calculations, including the mid-arm circumference, mid-arm muscle circumference, creatinine height ratio, and the creatinine height ratio is calculated as 24-hour urine creatinine multiplied by 100 over the expected 24-hour urine creatinine for height. This calculation results in a percentage that can indicate protein depletion. There are many methods of determining the body composition use the body weight as a measurement, determined via the weighing scale. Other details such as height and age can be correlated with other measurements and often used in the estimation formulas. So, validity about all these measuring or the assessment of body composition. So, the methods above are each valid and notable in providing a measurement that can be used to determine the true body composition of a tested individual. However, each method does possess its own individual limitations such as accuracy, precision or expense, and the combination of methods also has limitations. Often, the relative change from one period to the next is more important. If an individual can maintain all the factors as similar as possible, even a simple method such as weighing may give enough information to determine true change in the composition. Then types of exercises. The ideal percent of body mass which should be fat depends on the individual's sex, age and physical activity. For example, a female who is 30 years old will have a different ideal body percentage than the male of 30-year-old. Both have different amount of fats in their body. An athlete will have different ideal than a non-athlete, and it can depend upon the sport. The physical activities which can help decrease the fat mass and increase the lean mass or both are same for everybody. Aerobic exercise, also known as cardio exercise, which involves the heart, can decrease the fat. High intensity interval training in a particular helps the decreasing the visceral fat. Visceral fat is near the internal organs, while subcutaneous fat is just under the skin. The former is more tightly associated with poor metabolic health. Bone and muscle strengthening exercise, also known as resistant training, decreases the fat mass and increases lean mass at the same time, though it does better than the latter. In order to prevent injury from the repetitive motion, people should do some resistant training with different parts of their bodies on different days. So, exercising for at least 5 days in a week is essential to maintain a very good body composition, as all the experts say. So, today we have learnt all the types of assessment of body composition and how to maintain your body composition. Thank you.